Hello and welcome back to Excelsureacademy. So till now we have studied in the cell cycle the different phases of the cell cycle, wherein which we have talked about the interphase and the M phase. Then again in the interphase we have talked about the G1, that is the gap phase, S phase and the G2 phase, right? So next we also talked about the regulations of the cell cycle, wherein which we also talk about the MPF regulation, that is maturation promoting factor. So likewise in this lecture today we are going to talk about the M phase and what are the different regulations which is being carried out in order to carry out the cell cycle properly. So the events of the M phase. So it is the most important phase of the cell cycle. So during mitosis, which is also called the which is also called the nuclear division. So in this case, in this mitosis, what happens? The chromosomes they condense. So we know that they are in the chromatin fiber like this. They are present inside the nucleus. So once they are supposed to enter the mitosis, they will start condensing, and wherein which what happens? They finally form the chromosome. So the nuclear envelope will of the cell will break down. The cytoskeleton reorganizes to form the mitotic spindle, and the chromosomes move to the opposite poles. So what is happening? So we know that inside the nucleus, your chromatin which is present is finally forming a chromosome. This nuclear membrane is starts to disappear, and whatever the cytoskeleton. So we know that mitotic spindles are formed by the microtubules. Those are the cytoskeleton proteins which forms the microtubules, of which mitotic spindles will be formed, and these mitotic spindles will finally help in the movement of the chromosome into the opposite poles. And so we know that later we'll study that the chromosomes will be aligned like this, and mitotic spindles will be attached so that they will be moved in the opposite direction, and finally we get two daughter cells, right? Okay, so chromosome segregation is followed by cell division, which is also called cytokinesis. These are the basic events of mitosis, which include chromosome condensation, formation of this mitotic spindle, attachment of the chromosome to the spindle microtubules. So sister chromatids then separate from each other and move to opposite poles of the spindle, followed by the formation of daughter nuclei. So this is what we have just I have just mentioned now. Okay, so stages of the mitosis. So we are studying from our, like I can say from the 11th and 12th standard, that the mitosis has got the four different phases, that is prophase, metaphase, anaphase and telophase. So this we have seen during the our practicals also. So in the practicals, we have seen that onion peel will be taken, it will be stained with the saffron in, and right, it will be observed under the microscope. So there in which we see actually the chromosomes alignment, especially in the metaphase, where you can see it is properly aligned in the one single plane, right? So yes, so the beginning of the prophase is marked by the appearance of the condensed chromosome. So this is important to understand and remember also for one mark question, that the prophase is marked as the appearance of the condensed chromosomes, each of which consists of two sister chromatids. So we know that inside the nucleus, your chromatin fibers will be like this, loosely, openly kept, right? And slowly, as the prophase it is reaching, what happens? It will start condensing and forming the chromatids, the chromosomes. So these two are called the sister chromatids. Now, why two are there? Because M phase comes after the S phase. So we know that in the S phase, the DNA replication has taken place. And that's why you get two sister chromatids, okay? The newly replicated DNA molecules remain intertwined throughout the S and G2, becoming untangled during the process of chromatin condensation. The condensed sister chromatids are then held together at the centromere. So whenever we make chromosome like this, so here you have got the centromere. Okay. Now, which is a chromosomal region to which proteins bind to form the kinetochore, the site of attachment of spindle fibers. So finally, when the spindle fibers are formed, they are attached to the centromere. Okay. And these are the spindle fibers. So these spindle fibers will finally will try to pull the chromosomes or the chromatids into the opposite direction. So this is the entire M phase. The different stages of mitosis has been shown over here. So you can see that over here, the first one is the prophase. So as already mentioned, that prophase is marked by the condensation of the chromosome. So you can see over here two sister chromatids. So here we have taken actually two ends. So two chromosomes are there. You can see two sister chromatids are also there. Now then it is reaching the prometaphase. Now what is prometaphase? Wherein which these centrosomes will be present in the opposite poles and the spindle fibers will start attaching to the kinetochore. Okay. So kinetochore means they will be attaching to the centromere of the chromosomes. Now at the metaphase, what happens? They are aligned in the single line or in the single plane and to which your spindle fibers are attached. Now, next what happens in the anaphase? In the anaphase, these spindle fibers will start pulling the chromosome into the opposite direction. Finally, anaphase is followed by the telophase where what will happen is that it will start condensing the entire cell. So what will happen? Telophase will be marked by the appearance of the nuclear membrane. So as shown in the figure, you can see that the nuclear membrane starts appearing and they start forming the nucleus over there, right? And you can see that the spindle fibers are no more and only your centrosomes are present in the two opposite cells at the pole, right? Then the telophase is followed by the cytokinesis, wherein which you get the two daughter cells. Now, both cells are capable of going again back to the interphase and the cell cycle will be repeated if there is no DNA damage has taken place. So the centrosomes which had duplicated during the interphase, they separate and move to the opposite sides of the nucleus as already shown in the figure, right? You can see the centrosomes which was having the spindle fibers, they were present in the two opposite poles. Now there they serve as the two poles of the mitotic spindle which begins to form during the late prophase. Okay, so your spindle fiber starts forming from the centrosome. Now, in higher eukaryotes, the end of prophase corresponds to the breakdown of the nuclear envelope. The cell then enters prometaphase. This is the transition period between the prophase and the metaphase. So what happens in the metaphase? Metaphase, the spindle fibers will be attaching to the kinetochores of the chromosomes and they will align in the one single plane. So during prometaphase, microtubules of the mitotic spindle attach to the kinetochores of the condensed chromosomes. Okay. Now, again, this is the actually confocal microscopy and it is again showing the same thing. So these are the confocal images. Very beautifully they have shown with the all. So you can see over here that it is in the interphase. So what is happening in the interphase? It is going and your nuclear envelope is actually disappearing. And you can see over here that your chromatins are getting condensed slowly. And finally, what happens? You will see that they are converted, they are condensed to form the chromosomes. Okay. Now, what do you see over here is actually your cytoskeleton proteins. Those are your cytoskeleton proteins. And remember, there are three types of cytoskeleton proteins: actin, intermediate filaments, and microtubules, of which microtubules we are studying now as in the form of the spindle fibers. Now, if you see in the prometaphase, so we know the prometaphase is the transition period between the prophase and the metaphase. So you will see that it starts forming the chromosomes and they will be aligned in the one single plane. So you can see this one. These are the two centrosomes are present in the two opposite poles. Now you see that the chromosomes have been aligned in this direction. So finally, what will happen? They will start pulling towards the different or the opposite poles. Now, once it is in the opposite pole, slowly, the cytokinesis will take place and two daughter cells finally will be formed. So, the end of the telophase, it will be followed by the cytokinesis. Okay, I really hope this is clear to everyone. Now, the kinetochores of the sister chromatids are oriented on opposite sides of the chromosome. So, they attach to the microtubules emanating from the opposite poles of the spindle fibers. So, already we have discussed. So, you have got the chromosome and it is attached like this. So, you have got over here the centromere to which your spindle fibers are attached. Okay. Now the chromosomes shuffle back and forth until they eventually align on the metaphase plate in the center of the spindle. Okay. Now this has reached the metaphase. So it is in the one single line or the center of the spindle. Now the transition from metaphase to anaphase, this is also a very important step. So later in the next lecture, we will study about the checkpoints which is important to carry out for the transition from the metaphase to anaphase. So it is triggered by the breakage of the link between these sister chromatids. So as they show that it is not only the centromere which is holding the sister chromatid, so you have got the linkage like this. Let me make it clear once again. So you have got the sister chromatids like this. Okay. And one second. Okay. And you have got the links like this. So these linkages are actually holding your sister chromatids. Now, mitosis ends with the telophase during which nuclei reform and chromosomes recondense. Cytokinesis usually begins during the late anaphase and is almost complete by the end of the telophase, resulting in the two daughter cells. Now, these links are also important. They are called cohesins. We are coming to that. So we have entered into the mitosis, right? And we also know that it is triggered by the activation of CDK1 cycling the protein kinase, and that is your MPF. Remember this, in the previous lecture we have talked about that MPF is regulated by the polo-like kinase and the aurora kinase. So this is like a positive feedback. So your CDK1 actually activates aurora kinase, and aurora kinase activates polo-like kinase, which again activates the CDK1. Okay. So CDK1 cyclin B act as the master regulator of the M phase transition, both by activating the other mitotic protein, which are the aurora kinase and the polo-like kinase. Okay. Now, what happens when this MPF gets activated? So chromatin condensation starts. Yes, that's already been discussed. Now, in prophase, early prophase, so chromatin condensation starts. Nuclear envelope breakdown happens. Fragmentation of Golgi apparatus happens, and the spindle formation. So these are the four important functions of the MPF which is going on. So what is the first one? Chromatin condensation. So now how it is happening? It is happening because of the phosphorylation of the condensins and the cohesins. Now what are these proteins? These proteins are responsible for sticking your sister chromatids together. Okay, we'll study about these proteins just in the upcoming slides. Nuclear envelope breakdown. So I have mentioned before that these nuclear lamina are made up of intermediate filaments called lamins. A, B, and C. Now, what happens is that in order to break the nuclear envelope, these lamin proteins, the IF, the intermediate filaments are the cytoskeleton proteins, they need to get phosphorylated. So once they get phosphorylated, they lose their connection with each other. And finally, your nuclear membrane breakdowns. Now, fragmentation of Golgi apparatus, again, phosphorylation of the Golgi matrix proteins will cause the Golgi apparatus to break down. Then spindle formation, again the phosphorylation of centrosome, kinetochore and microtubule associated proteins. So all these are because of the functions of the MPF. Now, mitotic protein kinase of the aurora kinase, which is having the aurora A and B, and polo-like kinase, they are activated by the activity of CDK1, which is a signal that is to enter the mitosis. Okay. Now, the aurora and polo-like kinases function with CDK1 in a positive feedback. Just now I have discussed that CDK1 will be activating your aurora kinase will be activating polo-like kinase, and this in return will again be activating the CDK1. Okay. So the condensation of the interphase chromatin to form the compact chromosome is the key event in the mitosis. Now, highly condensed chromatin cannot be transcribed. So that's why the most of the transcription stops as the chromatin condensation takes place. So it is obvious, right? As already mentioned that most of the transcription takes place in the G1 or the G2 phases, which is required so that they can produce the proteins required in order to carry out the mitosis. Okay, so the first thing about the chromatin condensation. So chromatin condensation is driven by the protein complexes called the condensins. Okay, now this is important to remember. Chromatin condensation is because of the condenses, which are the members of the class of structural maintenance of chromatin. Please guys remember this. This is very important. It will come for 1 mark question. So condensants are actually the members of structural maintenance of chromatin proteins, and they really play a very important role in the organization of eukaryotic chromosomes. So you know that. So already in the nucleus, your chromatin was present like this. Now this has been condensed, highly condensed to form the chromosomes. Now, both condensants and the another family of SMC proteins called cohesins contribute to chromosome segregation during mitosis. Now, what is the function of cohesins? As I mentioned, in the chromosome, these linkages are called, or it is because of the cohesins. So they bind to DNA in S phase and maintain the linkage between the sister chromatids following the DNA replication. And that's why the different chromosomes do not get tangled with the other chromosomes. So the chromosomes or the DNA which gets replicated is along with linked with its daughter DNA. So as the cell enters the M phase, the condensers are activated as a result of the phosphorylation by the CDK1 and Aurora B kinase. Please remember this also, that the condensers are getting activated by the activity of CDK1 and Aurora B kinase. Now, even if you don't remember that it is phosphorylation, but please always remember that CDK1, that is your cyclin dependent kinase. Kinase means it has to be phosphorylation only. And in the cell cycle, every time any regulation is because of the phosphorylation or dephosphorylation, as I have mentioned many a times in the earlier lecture also. The condensers are then replace the cohesins along most of the length of the chromosome, so that the sister chromatids remain linked only at the centromere. Okay, so what is happening as it is progressing into the M phase, that is, it is reaching the metaphase. Now I want the cohesins to lose its activity, right? Otherwise, what will happen? I have my chromatids ready. If it is still, sorry, let me make it once again. So they are attached like this. You have got the centromere over here, right? And then you have got the cohesins like this. Now, as it is progressing, so these are your cohesins, which is keeping the sister chromatids linked. Now, as it is progressing into the mitosis phase, now this chromatid needs to go to this side and this sister chromatid needs to go this side. So at that time, I want this cohesin to be deactivated, right? So the condenser then induce chromatin condensation by forming DNA loops leading to the formation of metaphase chromosomes. So finally, what is happening is cohesin will lose its activity, and so that the sister chromatids will go into the different or the opposite directions. Here, so you can see the activity over here. So what is happening? So these green color things are the cohesin which is keeping your sister chromatids together. Now once it is entering into the prophase. See, you can see in the figure also. So these are your cohesins which is keeping your sister chromatids together. Okay. So sister chromatids are kept linked with the help of the cohesin. Now this is all happening in the G2 phase of the cell cycle. Now what is happening? Entering into the M phase, your cyclin, CDK1, Aurora B and polo-like kinase are activated. So now the cohesins will be removed, okay, and condensers will be activated. So this will be activated. Now if you see this figure over here carefully, you can see that the DNA has been linked like this. They are forming the loop-like structure. Loop structures are formed. Okay. Then again it is condensed in the metaphase. You can see how many loops are formed. So these are again forming the loops, and this stage is becoming the highly condensed form of the DNA. And of course, when the DNA is condensed form, none of the transcription activity takes place. Okay. Now, next thing. So, we talked about the condensation of the chromatin. The second thing is about the nuclear envelope, which gets breakdown. So, breakdown of the nuclear envelope happens by the nuclear membrane getting fragmented. NPC, that is a nuclear pore complex, dissociates. Now, in case of nucleus, let us say this is your cell. You have got the nucleus, outer membrane and inner membrane. Now, NPCs or nuclear pore complex are only way by which any material, let it be RNA or proteins, it is only by or it is only through NPC it is possible for any entry or the exit. So even that gets dissociates, and that's why your outer and inner membrane gets separated. And nuclear lamina depolymerizes. So you have got the lamin proteins A and C, which is forming together like this, and these are held together with the B. So totally they form the nuclear membrane. Okay. So the depolymerization of the nuclear lamina results from the phosphorylation of the lamins by CDK1 and cyclin B activity. So what is happening? You have got your A, C together. If suppose this gets phosphorylated, what will happen then? The association of the lamin A and lamin C is not possible, and that's why it gets fragmented. So phosphorylation causes the lamin filaments to break down into the individual lamin dimers. So you can see that you have got the nuclear lamina over here, the membrane entire is there, and these are your NPCs. Okay. Now what happens if you see very carefully, you have got these. So let us say that this is your lamin A and the other yellow color one is your lamin C. Okay. Now if they get phosphorylated at both the ends, N terminal and C terminal. So what will happen? Just because of the phosphorylation, the lamin dimers are not able to associate together to form the nuclear envelope membrane. Okay. This is also very important. So this is how it is arranged in the entire nucleus, and they are held together by the lamin B like this. So you can see lamin B holding like this. Okay. I hope this is clear now. So we talked about, so what happens when it is entering into the mitosis? The first thing was the condensation of the chromatin. Second was the nuclear membrane fragmentation. Third was Golgi apparatus fragmentation, and last was about the spindle formation. So this is the third one. So what is happening in the third? The Golgi apparatus and ER starts fragmenting, which may either be absorbed into the ER or distributed directly to daughter cells at the cytokinesis. So this is also important because once the chromosomes are being separated and two daughter cells are formed, each cell needs to have its own ER and Golgi so that they can also produce the proteins again required for the cell growth. So the breakdown of the Golgi apparatus into vesicle is again mediated by the phosphorylation of the Golgi matrix proteins by the CDK1 and polo-like kinases. Okay. Finally, the fourth activity of this CDK1 and cyclin B is the spindle fiber formation, which is nothing but the microtubules. Remember, microtubules are nothing but your cytoskeleton proteins. Now again, microtubules are made up of alpha and beta tubulin. Okay, please remember. Now, the breakdown of the nuclear envelope then allows some of the spindle microtubules to attach to the chromosomes at their kinetochores. So this initiates the process of chromosome movement that characterizes the prometaphases. Okay, so now what is happening? The proteins assembled at the kinetochore include aurora B kinase, which is involved in the interactions of the chromosomes with the microtubules. So these are the things you need to remember, even though we are talking only about CDK1, cyclin B, aurora kinase, and polo-like kinase. So now we saw that the aurora B kinase is responsible for the kinetochore attaching to the microtubules. Okay. So once the microtubules are attached to the chromosomes, microtubule motor directs the movement of chromosomes towards the minus end of the spindle microtubules, which are anchored in the anchored in the centrosome. Now here you need to remember that the microtubule motor proteins, there are two types are there. Okay. So you have got the kinesin, which moves towards the positive direction, and you have got dynein, which moves toward the negative. Now, what is the meaning of positive and negative? Here, positive means this end, and this is your negative end. Okay, so positive means where more and more alpha and beta tubulin protein monomers will be, dimers will be added, and minus means where it will be actually getting the depolymerization. Okay, so kinesin will move towards the plus direction and dynein will move towards the minus direction. Now the action of these proteins will draw chromosomes towards the centrosome, which is opposed by the plus end directed protein. So here they are talking about the kinesin. and by the growth of the spindle microtubules, which pushes the chromosomes away from the spindle spores. So this you can imagine, by taking the physics concepts of friction. So in order to move ahead, your feet is pushing the surface or the road in the opposite direction, right? So when you push it in the opposite direction, then only you're moving forward. In the same way, here also, the chromosomes are moved towards the centrosome by pushing the plus end directed motor proteins in the opposite direction. And that's how it is moving towards the minus end. That is, in this direction, it will be moving. So giving the force in this direction, moving towards the opposite direction. Now, microtubules from opposite poles of the spindle eventually attach to the two kinetochores of the sister chromatins, and the balance of the forces acting on the chromosomes leads to their alignment on the metaphase plate in the center of the spindle. So in the figure also, if you see, this is your cell actually. Here is your centrosome, and this blue color thing is your metaphase stage, that is your DNA or the chromosomes has been aligned. Okay, so this is the metaphase stage. So that's all about the M phase. So we have talked about what are the different regulations for the M phase, especially it is the CDK1, cyclin B, Aurora B kinase, and the polo-like kinases. So if you could remember, then it is very easy. I hope this lecture was useful. If still any doubts are there, you can please comment or mail me at excelsureacademy1228@gmail.com. You can also put your queries into the Telegram channel. Thank you.